Good afternoon. This is Edwina Tanner. I'm bringing you our World Harbour Seminar Series 9 broadcast. Today we've got Paolo Mancuso speaking to us from Bologna and Elisa Bone speaking to us about the Billion Oyster Project. Paolo will speak first. He's a research fellow at the University of Bologna and he works in environmental geology and biological sciences. Its research aims to understand the functioning of micro and macro marine benthic organisms and their interactions with each other in the inhabited environments with multiple stresses in order to suggest guidelines to mitigate the effects of coastal urbanization. So thank you for coming along today, Paolo, and I'll hand over to you.
Okay, welcome everybody. During this presentation, I will start with a brief introduction of the problem related to coastal urban sprawl. Then I will offer an overview of the Emilia-Romagna coast that is the area of our interest. And then I will show you the projects that we are carrying on and their preliminary results. And I will conclude with the final notes evolved from our results. So let's start. Nowadays, marine coastal environment has undergone profound changes due to severe urbanization and overexploitation of natural resources. More than 50% of shoreline is modified by the presence of different types of artificial structures. And with the huge amount of people living on coastal zones and the ongoing civil level rise, there will be a continuous need for coastal protection. Then there will be need for other artificial structures. Despite different cities are investing in architectural and engineering solution to create beautiful green spaces on coastal zones, this great effort or imagination and just right to the shore, without considering the marine environment as an integral part of the urban landscape. For this reason, there is a growing interest on ecological-based solutions that are aimed to design novel ecosystems in urban areas to improve not only the quality of the environment, but also the human welfare. Of course, to do this, we need to know what is the ecology and functioning of urban environments. And this knowledge cannot be extrapolated from natural systems. In fact, what we observe usually in artificial structure are the characteristics of this artificial structure different compared to natural environments. Usually we have a prevalence of vertical surfaces made with unnatural materials, and usually these areas are also high polluted. Then we need to build a specific knowledge about artificial systems. And this is the aim of the work group 2, Green Engineering, of the WHP project, which is aimed to explore the distribution and the effect of artificial structures in harbors, investigating the use of eco-friendly materials such as wood, shells, or rocks, and or the effect of the additional structural features mimicking the natural habitat, and also the effect of seeding of native habitat-forming species such as algae, or bivalves and corals. Okay, our work takes place on Emilia-Romagna coast that is characterized by different types of problems. This is a high populated region and also is one of the main holiday destinations, not only during summer but during the whole the year. We have also severe urbanization occurs also erosion of the coast, fragmentation and loss of native habitats, and also eutrophication, and of course also severe artificialization of the coast. The image that you see on the left show the distribution of the huge amount of artificial structures that are placed along the shore of the Western Adriatic Sea, the Northwestern Adriatic Sea. Despite these structures constitute the main hard substrate of the coast, they host mainly ephemeral and non-native species. Moreover, in general, we observed that these artificial structures host populations with the lower genetic variability compared to the natural habitat. Now we move to Ravenna Harbour, that is the place where we are doing our experiment. The harbour of Ravenna is one of the largest commercial seaport of the Northwestern Adriatic Sea. It is structured as a major canal port extending from around about 11 kilometers from the center of Ravenna to the tourist seacoast. Moreover, there are also two large converging jetties stretching for the coastline towards the sea for about 2.4 kilometers. That affect the transport of sediment, shaping also the nearby touristic beaches. Currently, inside the harbor, we are carrying on 3 projects. The first project aims to explore the distribution and underlying drivers of native and non-native species. The second is aimed to investigate the effect of a habitat-forming species on species diversity, richness, and biomass. And the third project is aimed to test the combined effect of substratum complexity and seeding of habitat-forming species on the macrobenthic communities along the harbor seawalls. Okay, we start with the first study. And this is on the left top view of the port canal that is called Candiano, that go from the center of the city towards the seacoast. And this canal is the core of the commercial and industrial activities. The aim of this study, or the first study, is to quantify the distribution of native and non-native intertidal mussels and oysters that grow on harbor seawalls, and test whether if they differ along the canal. Moreover, we are interested to test whether the distribution of adult is related to environmental gradient or to propagal pressure. This is the experimental setup. We collected the macrobenthic community on the harbor seawall in these 5 sites, and on both the exposure. And moreover, in each site we set up scoring pads to collect the bivalve settlers across times. As you can see, the center part in the central zone of the canal, there are no sites, and it was not possible for us to set a site inside this part because of the intense commercial activities. And here there are really preliminary results, and the results show that the abundances and distribution of native and non-native mussels changing along the canal or the canal port, and we observe an increase of non-native species, Meteorraster minimus, towards the inner part of the port, while we observe a positive trend for the native species. that increase toward the open sea. As regard the settlers, these are just data from 2 times, and we observe also in this case that there are different amount and distribution of settlers, and in particular we have a huge amount of settlers on the open sea near the coast, and they decreased towards the inner part of the port. I forgot to say that during this part about the settlers, we have some problem about the identification. For this reason, we are planning to use molecular techniques to separate settlers of different species. Now we move to the second project, which is aimed to quantify the abundances and the structure of meiofaunal assemblages associated to habitat-forming species, in this case to Mytilus gallus provincialis, and test if there were differences between different types of artificial substrate, concrete seawalls versus rock boulders, and different exposure aspect, north and south. This is the experimental design. We set up this experiment in the long jetties of the harbour, which are composed in part of rock boulders and also of concrete seawalls. We investigate also this across different time, and the preliminary result show that the meiobentic assemblage changes between concrete seawalls and rock boulders, and between also the exposure aspects. In particular, meiofaunal assemblages have a higher diversity of rock boulders compared to the concrete seawalls. And another important aspect is that this diversity is related to the abundances of mussels. As regards the taxonomic identification, these represent a major problem because identification based on morphology sometimes fails to identify these organisms at lower taxonomic levels. A possible solution should be the use of molecular techniques such as metabarcoding techniques. As you can see on the left, this graph shows the identification of meiofauna that we were able to do by a morphological approach. So we reached the family level for now. But for the same group in GenBank, we have data that go to the level of species. So then we are going to apply molecular techniques in order to obtain a detailed taxonomic information. Finally, we move to the third project, which is aimed to test the combined effect of the enhancement of substrate complexity and seeding of habitat-forming species on the macrobenthic communities that grow along the harbour seawalls. These are the hypotheses, and in particular, we hypothesize that structural complexity will affect the assemblages that grow on seawalls, and also that the seeded habitat forming will reduce the number of invasive and opportunistic species. And finally, that the survival of the seeded habitat forming will be higher on tiles with greater complexity. Okay, this is the sample design. This experiment is a part of a global experiment. Then we follow a common protocol. We set up the experiment on the seawalls, in the 2 sides of the seawalls of the harbor, the Ravenna jetties, and the substratum complexity was manipulated using tiles with different complexity, a flat 2.5 centimeter and 5 centimeter depth. provided, of course, by the Reef Design Lab. While as regards the habitat-forming species, we use oysters, and we attach juvenile oysters that cover about 35% of the entire area of the tiles. And these are the preliminary results about the cover of sessile assemblages. These are the main organisms that we found, and the non-multidimensional scale graph show a clear pattern of differences across time. So we moved from the first month to the 6th month. We have a changing on the cover, on the structure of the cover assemblages. The increase of substratum complexity, moreover, and the presence also of the habitat-forming species reduce the presence of the ephemeral algae such as ulva and cladophora. As you can see, considering just the structural complexity, we observe a decrease of ephemeral algae when we move from flat tiles towards higher complex tiles. While if we consider just the seeding effect, we observe that apart from the complexity of the tiles, we have also in this case a decrease of ephemeral algae. So the effect of habitat-forming species reduce the ephemeral algae. Moreover, the mortality of oysters on average were similar between the two sites, but different between position, between crevices and ledges, and at least on the south side. This is an ongoing experiment that we are continuing to investigate. And to conclude, these results are clearly very preliminary, and at this stage, it is difficult to draw a specific conclusion. Our aim here was to provide an overview of Ravenna Harbour and the project that we are carrying on with the aim to informing future eco-engineering intervention, aiming to facilitate target habitat for new species and associated ecosystem function. I would like also stress other important side benefit of this work. This experiment have offered a terrific opportunity to train a new generation of young students in the novel emerging themes of marine urban ecology and green engineering. Also, this work offers a terrific opportunity for public engagement. The experiments are located along the high used objectives of Ravenna, which are a pole of attraction for various activities for the local local population that attract not only local but also other people that come here in Ravenna. We are contributing to a global social survey that was designed with the Harbour project, World Harbour project, to understand how does the people use the harbour areas and what are their expectations of this environment. Okay, with this I finished my presentation, and thank you very much for the invitation.
Okay, thank you, Paolo. We have got a question here. From, let's see, the question is from Chong Hoi Lee, and he's asking, why is the abundance so high at S5? So I don't know if you can go back to S5
okay.
And I'll explain that.
We are talking about the first experiment. This? is this. Do you see my slide?
Yeah, you can see your slide.
Okay, of the settlers, of in general, of the mussels. Is not specified, but okay. Really, because on the seawalls of the jetties, there are really huge amounts of mussels. They grow not only just attached on the concrete surface, but they grow. They use themselves as a substrate. So other mussels grow to other mussels, and we have a multiple layer of mussels. And of course, this yeah, I think that this is due also by the condition, the environmental condition that allow the growth of mussels. Mainly we have we have in this part we have mainly mussels, so that cover all the artificial structure.
Okay, thank you. That's all the questions we have for now. So we'll hand over to Elisa in just a minute. Sorry, we'll just put Elisa on. She's back with us.
I'm just sharing my screen.
Okay, we can see that. So Elisa is a researcher from who teaches in Australia at the moment, but she has worked previously in New Zealand and the US. And her most recent role was with Columbia University in New York, where she was a lecturer, and also led a project that developed methods to assess the habitat quality of urbanized shorelines in New York Harbour. She's a senior consultant to the Billion Oyster Project curriculum and community enterprise for restoration science, working as part of the science team to produce harbour monitoring field protocols for schools and citizen science projects. So Elisa, thank you for coming and presenting today, and I will hand over to you.
Thank you so much for inviting me to speak in this World Harbour Project webinar series, and thank you to all who are joining us online as well. As we just heard, I'll be speaking about a project that I've been involved in over the last few years with the Billion Oyster Project in New York City. And so here are, for this title slide, we just have some of my details if you have any extra questions that come up during the webinar. So we may or may not know that the oyster was a really key species within New York Harbour and played a key role in New York's history and in New York's development as a colony. So the eastern or Atlantic oyster, Crossostrea virginica, was once extremely plentiful prior to European arrival in New York Harbour and the New York estuary. And Mark Kalansky, if you haven't read his amazing book, The Big Oyster, I suggest you do, and he stated the history of New York oysters is the history of New York itself, and we can really see that because it was a key trade item with other developing colonies. It helped New York develop its economic independence, and also, of course, formed a key food item for the native Lenape people of the region. Also, we all know that oysters perform those key ecological benefits such as water filtration, shoreline stabilization, and reef formation as well. But as the colony developed into what the city is today, and I've just shown here a couple of stills from the work of Eric Sanderson in Manhattan, where the top picture is a recreated pre-European Manhattan shoreline, and the bottom is obviously modern Manhattan shoreline. We can see that there's been a huge number of changes in how that city looks, including shoreline hardening, decreased permeability, increased urbanization, increased trade, and so on. And this has in part has led to a dramatic decline in oyster populations through overharvesting, decline in water quality, habitat loss, as I said, as well as the more recent issues of disease and climate change. And the two diseases that are really big problems over there are MSX and dermo. Okay, so we know that these oysters are a huge decline. We also know that there's an increasing importance of stable shorelines and natural shorelines that can enable the dissipation of wave energy at those shorelines. And we saw a very pertinent example of that in 2012 when Hurricane Sandy came through New York City, and was quite a devastating hurricane. And with climate change, we know that these storms and the intensity of these storms is going to increase. The bottom image there is Breezy Point in Queens, which was one of the hardest hit regions in the area. So there's a lot of increasing interest in how we might be able to restore those once really plentiful oyster populations in New York Harbour to bring back some of the ecological benefits that these populations provide. And one of the real driver of these efforts in New York Harbour has been the Urban Assembly New York Harbour School. This is a public high school that's situated on Governor's Island. It's been set up partly through foundation money, and some larger foundations such as the Clinton Foundation are providing some backing for it. And one of the neat things that it does is that it is a maritime focused public school, meaning that all the students who are graduate from the Harbour School are commercially qualified in one of several maritime skills, such as scuba diving, so they come out as commercial divers, they come out as ocean engineers or aquaculturists or boat builders and so on. So this is a really great resource for, and has formed a really great base for the restoration of, or the attempted restoration of New York Harbour oysters. And I'll just mention at the end here that one of the key springboards for the Billion Oyster Project was through the aquaculture program that was run out of the Harbour School with Pete Malinowski, who's one of the aquaculturists, now director of the BOP, and Murray Fisher of the New York Harbour Foundation. So springboarding off of that, the Billion Oyster Project, as you might imagine from its title, its goal is to seed 1 billion oysters in New York Harbour and 1,000 acres of oyster reef by 2030. And I'll tell you in just a minute how they're going with that and how far along to that billion oyster goal we've managed to get. So it has several different components, the main ones of which are shell recycling. So the BOP will partner with New York City restaurants and use those and collect oysters that have been used in those restaurants and use those clean shells for spat settlement. And you can see up in the top frame, this is a pile of reclaimed oyster shell that will be used to then settle spat. And so the aquaculture operations at the New York Harbour School enable the seeding of those spat and also the raising of the larvae prior to that, and the raising of algae to feed those larvae. Another component is the oyster gardening program, which involves local middle and high school students, and that is middle schools years 6 to 8 and high schools years 9 to 12 in a program, and here they are, some of them in the middle here, in a program that puts those seeded those shell with the seeded spat on top of that out into the harbour, and they monitor them over a period of time. The last component there is a reef construction, where the BOP will partner with regional research partners and land managers to start building some experimental oyster reefs. And this is a little bit small, this oyster reef design down the bottom here, so if anyone needs a better version of that, I can send that to you after the webinar. So there's quite a few components here that go into the BOP, and I'll just let you know how they're going with all of those different components. So by the numbers, essentially, we have 20 million oysters restored, so they're on their way to that billion goal, and gallons of water filtered. Of course, this is projected amounts given as the amount that an oyster is able to filter over its lifetime, pounds of nitrogen removed, and pounds of reclaimed and recycled shell. So it's quite a large operation, as you would expect, and at the moment we have, for the BOP generally, have 129 sites with oyster gardening out in New York Harbour. 1 acre of reef has been restored, and most importantly for this, the focus of this talk at least, is that we have quite a lot of high school and middle school students who've been engaged with this program. And I'll just mention at this point in time also that the Billion Oyster Project Schools program really seeks to involve students who are from disadvantaged communities, from disadvantaged schools, so-called Title I schools in the New York City region. And these schools are schools that where students come from low socioeconomic backgrounds and might and need additional assistance like free lunches and so on that are provided in those schools. So the recently expansion of the BOP has involved increased partnerships, increased efforts to partnership in 5 areas. One is oyster production, the aquaculture operations at the school, but also with oyster farmers in the region. The collection of shell from New York City restaurants is increasing. They're trying to get more of that happening. Increased reef construction, increased monitoring of reefs as well, and that also involves community partners. And then BOP schools and public programs. So I've got BOP schools there in italics because that is the focus of the project that I've been involved with. Public programs involve things like going to Billion Oyster parties and having lots of different fundraisers and concerts and things like that to increase the knowledge within the community of this particular project and to get people out to their shorelines, which they often don't do in New York City. So let's talk about the project that I've been involved with over the last 3 years, which is the BOP CCERS project, a rather wordy Curriculum and Community Enterprise and Restoration Science. So it's quite an ambitious project. It involves several different partners, as you can see down the bottom there, including the Harbour Foundation, New York City Department of Education, Pace University, the Lamont Doherty Earth Observatory, New York Aquarium, University of Maryland, and New York Academy of Sciences, among others. So this is an NSF-funded 3-year program that came under the STEM C section, so it's a science, technology, engineering, maths, and computer science section. And it was really focusing on embedding hands-on practical learning of sciences to the school curriculum and embedding that experiential learning into middle schools specifically. So it involves a number of components. We seek to train the teachers in harbour science and estuarine ecology and really embed that restoration ecology into the school curricula. So we involve a number of teacher fellows who are recruited from local New York City schools, and they attend seminars and training sessions, and they also collaborate to develop what we call a cross-disciplinary harbour literacy program. So curriculum items and lesson plans in ecology, other science, maths, chemistry, and so on. We also seek to connect the schools and the citizen scientists who are using these programs through a digital platform, which I'll explain in a moment. And for this project, we've expanded on the oyster gardening, which was really putting oysters out into the water column within a cage and monitoring their growth and monitoring their survival. But we've expanded that to look at a whole community analysis. We're looking at mobile organisms, sessile organisms, water quality and site conditions as well. So just to give you that in a diagram format, we have an approach that encompasses 5 resource pillars and 5 project settings. So the resource pillars being training the teachers, the student learning curriculum, the digital platform where everyone can look at those resources, after school and summer mentoring and community restoration based exhibits. So that would be partnering with the New York Aquarium and other organisations. And 5 project settings for that: the PACE classroom for the teacher training, the New York City Department of Education to school classrooms to teach the students, the waterfront sites for field research, after school programs through the Good Shepherd Services and Marine Science Research Facilities at New York Harbour. And I've got in a yellow box there the digital platform and the waterfront sites for field research, because that's what I'll really be talking about today. How we develop field protocols so that students and teachers can monitor their estuarine sites and monitor their oyster growth and get engaged in hands-on practical science. Okay, so how are we going with all of this? So this is at year 2, we're currently in year 3 and we're winding up. But back at year 2, we had 65 teacher fellows from 35 middle schools across the region and engaged 6,000 students within the program. Field season 3 is commencing right now because it's summer in the US. We finalised those field protocols. We finalised ID guides, species ID guides, and we're developing field materials for web and print. We have a mobile app that we developed in season 2, but that has been succeeded by the new BOP digital platform. And I'll talk about those 2 items as well and how that allowed us to engage teachers and students in field monitoring. Aquarium exhibits are still in development for the New York Aquarium and for the River Project. And if you want any more information on that, you can have a look at that link down there, which will be provided. This photo at the top here is one of some people from the River Project who are training some teachers in species identification in New York Harbour, and down the bottom here we have some students attending the River Project exhibits. In the middle, this is the front page for the app, the mobile app that we were developing for this particular project. Okay, so I just want to get into what we actually did in terms of monitoring. So we developed 5 separate protocols that teachers would carry out with their students. So one protocol would monitor site conditions, things like just weather variables, shoreline variables such as shoreline type, shoreline slope, and things like that. The second was oyster measurements. Sorry, I'll go back a little bit. So we actually designed a specific purpose-built portable device so that all these teams would be able to go out and put in their local harbour site and bring it back up and monitor periodically. And this purpose-built device included oysters for monitoring, spat on shell for monitoring, but it also included things like settlement plates and a compartment to capture mobile invertebrates as well. So we had it was an all-in-one device, and again, if anyone needs any more information on that, I can send that on after the webinar. But in the interest of brevity, I'll just go through how we engaged them in a more broad sense. So we had site conditions, oyster measurements, so we measured oyster growth and survival, and we measured mobile organisms, so organisms that were captured within our colonisation device. We measured sessile organisms, those that were settled on the settlement plates, and also water quality, some really broad basic measures of water quality that were suitable for middle school students and their teachers to carry out. We created sets of instructions that were suitable for those large student groups, and so teachers had the option to split their groups up into 5 groups to match the protocols. But there was also a modular protocol set that groups that were larger or smaller were able to adjust as they saw fit. So in year 2, we created this mobile app, and you can see some of these screenshots here up on the slide. That was really user-friendly, very interactive interface, and lots of pictures and lots of big icons for the teachers and students to see. That was the primary mode of data collection in year 2, and although it had the capacity to produce those really nice, robust data on community monitoring, we had some technical difficulties in the field when we were utilising that mobile app. So that had since been succeeded by the digital platform. Okay, so this has been succeeded by our BOP CCERS digital platform, and it's available at a link up there, and anyone can actually join in that to become a team leader or a team member. Although if you're in New York City, or if you're close to New York City, if you know somebody in New York City, I encourage you to sign up and go to a training session and help this restoration effort. You can see this is the sort of the screenshot of the front page of the digital platform, and it's quite user friendly, and it's very has those very broad numbers here. So at the moment, at this particular time, we had 456 team members, 400 team leads, 449 teams, and 250 something organisations as well. So we're getting quite a lot of uptake of this particular program, and the data platform has been a really useful tool to have everything in one place. It's really a one-stop shop for these user groups. So it combines the data collection and the storage from the different expeditions. It also combines curriculum and lesson plans, research findings. An events calendar is embedded. Participants can partake in forum conversations with other participants as well, and it also offers those detailed site metrics. So who's coming to the site, who's using it, what are they doing in the field, and how many expeditions have we had so far. So you can see down the bottom here on this map, this is the sites of the different expeditions or the different colonisation devices called oyster restoration stations that we have across the region, and it's growing. It's obviously concentrated in Manhattan, but we're growing out to other regions as well. So far we're really getting some pretty good feedback on the participant experience, and we're getting a really great uptake on participant groups. So we're getting not just schools, but it's also broadening out to community groups, businesses, and also government groups as well. I'm just going to show you a couple of screenshots from the digital platform. So this is, as you can see, we've got a few different tabs here: community, curriculum, restoration, and events. This is just a summary of some of the site metrics here. So we can see that at the moment we have 132 oyster restoration stations. That is the project name for the colonisation device that I described before. We've had 226 expeditions, and these are the ways that they change over the time. You can see that we probably have fewer expeditions in the northern winter, as you'd expect, and then it picks up in May and June. We can see which stations have been lost, which stations are active, who's completing protocols, who's not completing protocols. Obviously, we've got a lot of, still have some problems with people not completing the protocols and not completing the expeditions, but this gives us a good overview of where we should target our efforts to improve the protocols themselves and the interface with the participants. Just here I've got a screenshot of one expedition that someone's come back with, and this is how the data would be uploaded. This is just a data visualization. So it can be downloaded into spreadsheets, but for the interface that we have here in the digital platform, it's quite visual and it's quite interactive here. So you can see that the participants have been asked to upload photos of the different oysters that they've got here and upload a photo of their oyster cage, so that we can look at how much it's been, like how many organisms have accumulated on the side of that, for instance, and we can have a look at the condition of these particular spat-on shell. So we can provide some feedback on the data they're collecting. And we also have, pardon me, we also have many inbuilt limits on the different fields and inbuilt alerts to alert participants if their parameters seem like they're not within the range that we expect. Okay, so summing up, we're generating some useful data on the oyster monitoring process. We hopefully will start to generate some really robust data on biological monitoring, biological populations of sessile and mobile invertebrates. But we're really getting a really nice uptake from the community and a really nice spread of groups. In terms of, we have community groups, as I said, businesses and government, as well as schools. So the word is getting out there, and we're getting a lot more groups participating in this project, which I think is one, is good for their education, and two, is good for the harbour as well. So right at the moment, as I said, we're summing this project up. We're completing the training of the current cohort of fellows. We just had our annual symposium on June 23rd. Obviously, I'm not over there to participate, but I hear it was great. This year, we'll be working on disseminating our outcomes. We'll be disseminating outcomes on how the protocols went, how they performed, how our data collection procedures performed, and how the teachers and students engaged with the program and how it improved their learning in sciences. We're hoping for some sustained fellow involvement with the Billion Oyster Project, so the teacher fellows that we've recruited over these 3 years will stay on with the BOP and stay on with the monitoring programs, and we'll recruit other teachers within their networks, schools and networks to the BOP as well. And one thing that we're always thinking about is how we could improve the community involvement and improve those field protocols and trial them in different areas as well. So we're exploring some options there, and I'm happy to speak to that in the questions as well. So I just wanted to finish up with a couple of testimonials and a couple of pictures to show kind of the joy that people are getting out of this project over in New York City and how it's really engaging students that may or may not have even been to the water before. And personally, we had some great experiences over there, and it was so good to see some of the students get excited about this stuff. I have a small presentation at the end here. I don't know whether we have time for that, but I will just ask that question, and if we do, I can put that up, and if not, we'll take questions now. So let me know if we have time for that.
We have time for that.
All right, I'll put it on then. I apologise if there's any feedback at all, and hopefully it will run fine. Now.
There is a little delay.
We're not getting the sound through, Elisa. So,
Oh, I'm sorry. Okay. It's coming through to my headphones, but hang on. Okay. Maybe I can leave that there for people to look at after the fact.
okay, we have the web address there on site. Thank you for that. You can leave it scrolling if you want, because there's no sound. We can ask questions.
Okay.
Yeah. So they can just see. It looks good.
Sorry about that.
So we've got a few questions here from Chung Ho Lee again. He's got a number of questions, and his first one is, what is the material of the oyster cage, and how many oysters are put in each cage?
Okay, so I'm just going to mute this video at my end so I'm not distracted. Okay, so the oyster cage that we were using was a steel mesh, vinyl coated steel mesh. It was pretty readily available from commercial fisheries shops and fishing shops and things like that. So it was really, and what we were doing was we also created a protocol for how to construct that oyster cage. So we trained the teachers in oyster cage construction, and it was a really neat, it was another way to get them engaged in the process and get the students interested as well. So the oyster cage itself is made out of a, as I said, it's vinyl coated steel mesh. This was based on some of the work that we did on the shoreline habitat quality assessment, which was work that I did previously. As you can see on the screen there, oh, you can't see it now. Okay, so it was vinyl coated steel mesh, and then we also had a bird netting, sort of a insect netting that would trap algae and sediment and detritus, and that would form a substrate for mobile invertebrates, and that was quite successful. we also had settlement tiles that were made essentially from ceramic tiles found at hardware stores, so unglazed ceramic tiles. And all of these materials were designed to be easily found, reasonably cheap, and easy enough to put together for the non-specialist.
And how many oysters were put in each cage? About?
We had 50, and then there were 10 that were tagged. So we had 10 that were tagged, and then 40 others that weren't tagged. And the participants are asked to monitor all of the spat on each of those tagged shells. So they would take pictures of those tagged shells, and you could see the location of the different spat. And then there was we had specific instructions on how they would do that and how they would work their way around the shell, so that they wouldn't double count any spat.
Sure. And what species and how many species were used?
We just used eastern oyster, Chrysostria virginica, and that was it. That was the only species that we used for oyster.
Okay. And are these oysters going to be harvested at all?
No. This is not a. They're not for consumption, human consumption. Yeah. They're just for restoration, just for ecological benefits. And I guess I'll add at this point that the oysters within the oyster gardening and monitoring exercises stay in their cage, but once. So of course they're not contributing to reef development, but the oysters. We also have additional oysters that are used for reef development as well. So this is about how well they could survive in New York Harbour and how well they can grow, as well as how well those associated organisms are developing and those associated communities are developing and surviving as well.
How long are the cages deployed in the sea or harbour? How long are they in the water?
So it really depends on the participants themselves. We recommend that they stay out there all the time and they're monitored at least once every 3 months. So we want people to go out on monitoring excursions at least quarterly. It really depends on the participants and how they're going with it, logistics and their timeframes and their scheduling as well as to how often they're getting monitored. But we do like to have them out continually if possible.
Just to finish up from Chung Lee,
sure.
He said, thank you very much for sharing, and it's very interesting on how the community is engaged in this project. You've really managed to engage them, so thank you for sharing. The next question is from Andy Meyer. He says, you often see projects in the US using plastic mesh in oyster reef restoration projects. Are you of any trials using natural fibres?
Hi Andy, how are you? This was really out of my hands in a sense. It was really, we started off with the BOP oyster garden, which was a steel mesh construction, and then we added on these different components to do mobile and sessile invertebrates. And as I said, it's not a restoration project necessarily. It's more of a monitoring project to monitor colonization and so on. So these cages aren't going to be left there in perpetuity to provide a substrate for restoring a reef. They're there as an engagement and an education tool, and also a data collection tool. To speak to the second point, whether they're going to involve natural materials, there's a lot of work. I'm less involved with the reef restoration side of things for the Oyster Reef Restoration Network, but they do use concrete and other constructions like that, but they also are looking into natural materials as well. And I know there's a lot of great work being done on that, both here and elsewhere. So thank you, Andy, as well.
We'll leave it there. We've done very well, and thank you very much to both our speakers, Paolo and Elisa, for sharing your wonderful presentations with us today.
Thanks very much for having us.
Thank you very much.
Okay, goodbye.
Goodbye. Bye.
Bye.